Hey all, so we're going to be looking at protein synthesis for this video, and we are going to be trying to figure out how a cell produces a sequence of amino acids from DNA, and also how is the reliability of protein synthesis ensured. So we're going to be trying to think about continuity and change. Again, how are we keeping things the same, like the DNA, and coding effectively for RNA, but then simultaneously making new things like proteins. So transcription is the process of making RNA, and RNA polymerase is the thing that's going to do all of that. And so DNA is used as a template, and RNA is going to build onto the 3' end of the template strand of the DNA. And RNA polymerase is going to unwind a segment of DNA, not the whole thing, just a piece, which is going to be the gene that's going to be copied. And RNA polymerase is going to make covalent bonds between the sugar phosphates on the RNA nucleotides, and it's also going to detach RNA from the DNA strand. And remember that in RNA, there's no thymine, there's only uracil. And so when we're thinking about the template strand here, T's are going to make A's, and then A's are going to make U's, not T's, so uracil here. And RNA is then going to move out of the nucleus once it's detached from the DNA, and DNA is going to reform its double helix quickly, so it's going to prevent mutations. And then once RNA is into the cytoplasm out of the nucleus, it's going to go through the process of translation. And so translation is where the RNA connects to the ribosome and it starts to make a protein. So first off, the RNA is going to connect to the small subunit of the ribosome, and then the large subunit is going to bind onto there, and it's going to start to read the code from the RNA. And the large subunit has 3 catalytic sites. Those are called the E, P, and A sites. So E is for the exit site, P is peptide bond site, and A is the arrival site. And the ribosomes are going to be read from a 5' to a 3' direction, meaning the amino acids are going to be added on to the 3' end from the transfer RNA. And you'll notice here on the transfer RNA, there's 3 little letters on there, and then they match up with the 3 little letters on the messenger RNA. So those 3 letters are called a codon. And then the 3 letters on the transfer RNA are the anticodon. And that anticodon is specific to one amino acid, just like the codon is specific to one amino acid. And the job of transfer RNA is to bring that amino acid to the A site. Then it's going to shift over. It's going to make a peptide bond with the next amino acid in the P site, and then it's going to exit. And so only 2 transfer RNAs can be in the catalytic sites at the same time. So codons are a series of 3 bases that code for one amino acid. And you can see here, if you're looking at messenger RNA, you would go, let's say it's UUU, that's going to give you this amino acid, which is phenylalanine, but you would never need to know that. Either they're going to tell you the names of them, or that you would just have to identify that PHE is the amino acid that would be coded for by UUU on messenger RNA. The codon on messenger RNA binds to an anticodon, and so you can see here some representations of the structure of transfer RNA. And so this is actually more what it looks like, but this is a more stylized, easy to visualize version. And those are both molecules of transfer RNA with the amino acid on the top and the anticodon on the bottom. And transfer RNA picks up amino acids using the enzyme transfer RNA activating enzyme to signal to the transfer RNA to pick up an amino acid. And the amino acid actually binds onto the 3' end of the transfer RNA also. So when we're looking at reading of messenger RNA, there's always going to be a start codon and a stop codon, and that signals to the ribosomes to start reading and then to detach the messenger RNA from the ribosome. And once the messenger RNA is detached from the ribosome, it can be picked up by another ribosome, or alternatively, if enough proteins have been made, then the messenger RNA in the cytoplasm will just get broken down and no further proteins will be made. So some mutations can occur, and we'll look more at this when we're talking about genetics, but some mutations can occur to the sequence of DNA bases, and that can cause significant problems for the making of proteins. An allele is just a different variation of a gene. So what that would mean is that maybe it has one or two bases that are slightly different, and so the protein looks slightly different. And often this has no significant impact. It has a neutral impact, but at times it can have quite a detrimental impact. Sickle cell anemia is kind of a middle of the road impact. And so mutations are occurring randomly all of the time, but a base substitution mutation is what's going on in sickle cell anemia. And so what's happened here is the T base has been replaced by an A base. And so when the messenger RNA is made, it goes from GAG to GUG. And this codon, GUG, codes for an amino acid valine, but GAG is going to code for glutamic acid. And as a byproduct of that, glutamic acid and valine have different structural components. And when valine is introduced into red blood cells, it makes the red blood cells lose their circular shape, and instead it becomes like a crescent moon shape. Crescent moon shapes, if you have some of them, actually are very useful because malaria, which is transmitted by mosquitoes, it's a parasite, it infects red blood cells. And if they have that crescent moon shape, the parasite isn't able to enter into the red blood cells. So that said, if they have that crescent shape, they can also get caught in your blood vessels. They can, so a normal red blood cell has a lifespan of about 180 days, but one that has sickle cell anemia can have as short of a lifespan as 4 days. So your bone marrow is always trying to make new red blood cells. And so sickle cell anemia has an evolutionary advantage if you have one allele version, but not if you have both alleles. If you only have sickles, then it's almost impossible for you to breathe and transport oxygen through your body. Additionally, you make clots, and then you throw clots, and then they go to your brain or your heart, and then you can die. So to summarize this entire section, messenger RNA is read by ribosomes to create proteins from amino acids, and we just need to know how that happens. Okay, thanks for watching this video.